Hello everybody. Today we're going to be talking about the chemistry of life. And this one's really interesting because we get to talk about what we put into our bodies. Really interesting because a lot of people don't actually know the chemistry behind what they should and shouldn't be eating. So we get to dig deeper in this one. So let's get started. So first things first, let's get started on talking about what we're going to be covering today. So we're going to be talking about atoms, which are the building blocks of molecules, and then we're going to be talking about the chemical bonds that atoms are involved with. There's 4 different kinds of chemical bonds. There's ionic bonds, covalent bonds, hydrogen bonds, and van der Waals interactions. Okay, 2 of these are a lot stronger than the other 2. Okay, so 2 of them are going to be considered strong bonds, and 2 of them are going to be considered the more of the weaker bonds. Life is made up of matter, and matter occupies space and has mass. Okay, matter is composed of elements, and what is an element? Okay, elements are substances that cannot be broken down or transformed chemically into any other substance. They are going to have those chemical properties of those specific atoms only. Okay? So atoms are small components of an element that retain all chemical properties of that element specifically. So elements, so let's say hydrogen, for example, and that's the example I have down below. Hydrogen atoms have all the properties of hydrogen. So all of the atoms of hydrogen are going to all have and be gas at room temperature. No matter what, they're all going to have all of the properties of those hydrogen atoms at room temperature. So they're going to maintain those chemical properties of hydrogen. All of the atoms are going to do it. So they're going to maintain all the chemical properties of hydrogen, as would gold. Gold atoms would maintain all the chemical properties of that gold element as well. So let's start off by figuring out what makes up an atom. Okay, atoms, in the simplest form right here, are made up, first of all, of protons in the nucleus as well as neutrons. As you can see right here, protons are positively charged. Neutrons are neutral. They are not charged at all. Electrons, which are on this orbital shell on the outside, are actually negatively charged. Okay, so protons positive, electrons negatively charged on their orbitals, and then neutrons neutral charge. They don't have any charge at all. Okay, so this looks like a lot, but we're going to go ahead and break it down to make it very, very simple. Okay, atoms, remember, multicellular organisms are made up of a combination of elements. So these elements contain, remember we went through this in the very beginning, the whole level of organization. Atoms combine, they make molecules. Molecules interact, they make cells. Cells combine, make tissues. Tissues combine to make organs, and so forth until an organism is made. So remember right now, this past slide, we said atoms are made up of, which ones are the ones that are positively charged? Protons. Okay, protons positively charged. Then the neutrals, the neutral atoms that are inside the nucleus, right here, those are the neutrons. Then the ones that are negatively charged, the ones that are outside in the orbital of the atom, those are your electrons. Okay, there is an exception. Hydrogen is an exception to this whole rule. Okay, hydrogen has 1 proton, 1 electron. Okay, there are no neutrons right here in hydrogen. That is the one exception to the rule. Now, the elements, we're going to see what it looks like when you're looking at that whole huge periodic table of all those elements. Okay, when you look at that periodic table, this is what you're going to be looking at. We're going to kind of break it down So when you look at that guy, you kind of understand what you're looking at. The atomic number, the very, very top, is going to be, actually, that's telling you also how many protons there are. So number of protons is also the atomic number. So if you're looking at carbon, the atomic number is 6. Guess what? That's also how many protons there are. So right here, carbon, 6 protons. Go back. Okay. Mass number, or the atomic mass, that is actually the number of protons and the number of neutrons. Remember, neutrons are neutral. Okay, so atomic weight right here, atomic mass, that's protons. Remember, those are positively charged, and the neutrons. Okay, so that is going to give you the atomic mass. That's going to be right here. And the electrons in neutral element is also going to be the number of protons. So that's a lot. They give us a lot of information just here. So if we're looking at this example right here, so carbon, this right here. Carbon has blank protons. So we already learned atomic number is also equal to protons, so we know 6. We would write 6 right here. The number of electrons, well, it did tell us electrons in a neutral element is also equal to the number of protons. So the number of electrons would also be 6. And the number of neutrons. Well, this is a little different. So, number of neutrons, well, we know that the mass number down here is equal to protons and neutrons. And the protons are up here too. So what we can actually do is the mass, total mass, minus the number of protons, and that would give us the number of neutrons by themselves. So I could do 12 minus 6, and that would give me the number of neutrons, which is 6. So if you ever didn't know the number of neutrons, you could always do mass minus protons, and that'll give you the number of neutrons. Okay? So if you ever didn't know, you can always figure it out by looking at the periodic table to find out with any of these. So another example, let's do helium. The number of protons would be 2. The number of the atomic mass would be the 4 underneath the atomic symbol, which is He. Okay? And if we wanted to find out the number of neutrons, all we would have to do is atomic mass, which is 4, minus the proton number, which is 2. So 4 minus 2, and that would give us the number of neutrons, which is 2. Pretty simple. And once again, this looks like a lot. I will break it down. Do not worry. This is kind of like a cheat sheet that I tell all my students to use. Okay? So, cheat sheet. These guys over here, these over here on the left-hand side. Okay? These are going to be your periods. Period 1, 2, 3, 4, 5, and so forth, all the way down to 7. Okay? These are going to give you a little cheat sheet to tell you how many orbitals there are around these atoms. So, very, very simple. And what I mean by orbitals is how many actual orbitals there are going to be around the nucleus. If you ever have to draw them to figure out how many valence electrons there are. Okay, valence electrons are the electrons on the outermost ring of that atom. That's very important because those are the only electrons that are going to react with other atoms. Okay, so let's go ahead and draw lithium, for example. Lithium right here. Okay, I'm going to get my pen. Okay, lithium. Let's go ahead and look at this. Like I said, this is my period 2. So here's my nucleus. My nucleus has protons, remember? So protons are positive, so here's my 1 positive, my 2 positive. I'm just going to draw. It's hard to see, but here's my 2 positive protons, okay? And I'll go ahead and draw. I'm going to do magenta for my neutrons. They're just neutral, so they're not going to get any kind of charges. They're just in there, okay? So here is, actually I need 3, sorry. Actually, let me get my third. There we go. And I need one more proton in there. Come on. There we go. So 3 protons, 3 neutrons, okay? Now, this is what I was talking about by a little cheat sheet in here. Here is, let's do red. Okay, my orbitals, I'll do in red. So here's my one. It's telling me I have 2 orbitals to draw. So here's my 1 orbital around the nucleus. Here's my 2 orbitals around the nucleus, because that's how many it's telling me. 1, 2. Okay? So these are telling me how many orbitals there are. And on the first orbital, you always only have 2 electrons. Second, third, fourth, fifth, sixth, seventh orbitals, there's always 8 electrons. But since we only have 1, 2 orbitals, obviously, that's all I can draw. So, lithium has 1, 2 orbitals, and obviously, all I can draw is 3 total electrons. So, here's my 1, 2, and 3 electrons. I'm done. Simple. Now I'll do my little negative charges. And I'm drawing the atom of lithium. But like I said, this is very simple because this is telling you how many orbitals you would have to draw if you're drawing the atom. I love it. Another way to cheat, if you're trying to draw the atoms quickly, efficiently, getting you through the test rather fast, is looking at the group numbers. The group numbers up here, 1, 2, and if you're going 3, 4, 5, 6, 7, and 8, is these group numbers up top are telling you how many valence electrons to draw on those orbitals. So, once again, these periods on the left are telling you how many rings or how many orbitals to draw around the nucleus. And then these groups up top, if you ignore these transition metals, these guys are telling you how many valence electrons should be in this orbital that you're drawing. So remember, we drew 1, 2 rings or 2 orbitals. And then guess what? This group number 1, because that's where lithium was, told me how many electrons should have been in that orbital. Because if I had to draw carbon right here, this would have told me, oops, sorry, let me go back. This would have told me right here that I would have drawn, let's say this is carbon. That means I would have had to draw, since it's right here in period 2, 2 rings or 2 orbitals, and then 4 valence electrons. That would be so fast, so simple, I could draw it instantly without even thinking. Real cool way, quick way to not even think about it and get through the exam rather quickly and the question rather quickly. So real good cheat way. Not even cheating, it's just a real quick way to get through it without worrying about valence electrons and not worrying too much. You just know stuff, get through it. Great way to understand the material. Okay. Like I said, it's really important to understand what valence electrons are, because valence electrons are basically going to tell you whether an atom is going to react with another atom or not. Because if that outer shell is full, because if all 8 electrons are there, it doesn't want to react with anything else. It's happy, it's full, it doesn't have to share, it doesn't want to share anything. Also, it doesn't want to take anything else from another atom because it doesn't need to. It's content with its full outer shell. It's good to go. Okay. If also, let's say, as you guys can see here in group 1, if it only has 1 valence electron, it does want to share. It has that one lonely soldier. It wants to give up that one lonely soldier, and it's very, very reactive. Same thing as you guys can see in group 18, way over here. Group 18, like I said, it has all 8 fully filled valence electron shells, it's rarely going to react. That's why I highlighted it in red. Rarely going to react. It's happy. It's not going to share, and it's also not going to take any extra electrons. Okay, that's why I highlighted both of those, so you guys can look at that and try to understand that a little bit more in depth. Okay, so isotopes. Isotopes are going to have the same number of protons like we've been talking about, but here they're going to have a different number of neutrons. So instead of having 6 protons, 6 neutrons, like a normal neutral atom would, this is where things change. A lot of people, you've probably heard of carbon-14. A lot of people have talked about it. That's a common one a lot of people talk about because that's actually one that archaeologists use a lot of the time for carbon dating. Okay, that's what we use to identify how old fossils are. The reason being, scientists are able to identify at a specific rate that carbon-14 decays. So we can actually follow carbon-14 and its half-life and say how old an item is because of it. So it's really cool because that's how we can actually say how old a person was at a certain era or a fossil and so forth. So really neat, really cool. But that's what an isotope is. It's an atom that has the same number of protons but a different number of neutrons. Okay. So like I said, this is an example of archaeologists actually using carbon-14 to age the remains of a pygmy mammoth here. This is exactly what we've been talking about as far as the valence electrons. It's going to determine how the elements actually interact with one another. Okay, like I said, the first energy level or the first orbital holds up to 2 electrons. All the energy levels after that are going to hold up to 8 electrons. So some of the atoms are going to give up an electron. Some are going to accept an electron. That's going to be when they're actually going to be called something different. Whether they give up or gain an electron, they're going to be termed as ions. Okay. If they lose an electron, they're going to be considered a cation. If they gain an electron, they're going to be called an anion. Okay, and that's what you guys can see right here. Lose an electron, cation. Gain an electron, it's an anion. And this is what I'm talking about right here in this picture. This sodium chloride ion that is formed, this is your common known table salt. This sodium ion actually has this one lonely soldier out here. This one electron wants to give up this electron to this chlorine ion in order to fulfill this 8th electron so that it can be a full 8 electron and fulfill it over here on this chlorine atom. That way, it'll be an empty ring over here in this orbital on the sodium atom, and then it'll be a full orbital over here in this chlorine atom. That would make it a sodium chloride compound altogether, but at the end of the day, this would make it a cation, and this would become an anion. And this is just basically breaking it down all together so you can see it from the beginning to the end, and so you can understand cations lose, anions gain electrons. The example is actually in your book as well, so you can look at it a little bit further. And this is a quick quiz as well, so you can test yourself a little bit. You also do want to understand this a little bit further as well, because you will be going through these items for your lab as well. So Another thing you were going to want to identify is the fact that you are forming, not only are they becoming ions, this is also known as an ionic bond. So like I said, electron is being given away, one is accepting an electron. It's not only forming ions, they're also forming an ionic bond. Just because one's giving, one's gaining an electron. So just a different way of viewing it. Covalent bonding is the strongest of all of the most common forms of chemical bonds in living organisms, because these guys are not only giving and gaining an electron, they're actually sharing their electrons here. So not one's gaining, not one's giving, they're actually sharing them and mutually here. So as you guys can see here in the hydrogen gas, they're mutually sharing their electrons. And we have a structural formula down below where you have a hydrogen and then a line and then a hydrogen. That's actually, that one line is showing that, that chemical bond between them. And a molecular formula of that would be the H with the subscript of the 2 down below. And there's two types of covalent bonds. There's polar covalent and nonpolar covalent. So polar is when the electrons are shared by the atoms, but the electrons actually spend more time around one nucleus than the other. So in that good example would be around the water molecule. The electrons actually spend more time around the oxygen nucleus than the hydrogen's nucleuses. It's a really, really good example. You're actually going to refer back to water being a polar molecule a lot, especially in labs that you're going to be seeing. And then the other type of the other type of covalent bonding is nonpolar, and that's between two atoms of the same element or between different elements that share the electrons equally. So those electrons are actually going to be going around the atom's nucleuses equally, and they're not going to be spending more time around one nucleus than the other. So these are good examples. Like I said, over here on the left-hand side, you'll see the water molecule. So as you can see with my pointer, it is going to be spending a lot more time around the water molecule, around the oxygen's nucleus than the hydrogen's, and versus over here on the methane molecule, they're actually going to be spending an equal amount of time around the nucleuses of all the different atoms here around the oxygen molecules, as well as the methane molecules right here in the middle as well. So these two would be considered nonpolar, whereas the water molecule would be considered polar. And this is going to be considered a hydrogen bond right here, which is considered a weak bond. It's slightly positive hydrogen of one water molecule attracted to the slightly negative oxygen in another water molecule. Like I said, this is considered a weaker bond. You can actually find hydrogen bonds in our DNA. The liquid nature of water is caused by the hydrogen bonds between water molecules. Hydrogen bonds give water the unique properties that sustain life. No hydrogen bonding at room temperature, water would have a gas rather than a liquid though. Really interesting. Water has a lot of unique properties. Hydrogen bonding does not have to include water. Hydrogen holds a DNA double helix structure together. That's the backbone of DNA, like I just mentioned. And it's also responsible for the 3D structure of proteins. And the last one we're going to be talking about today as far as the different chemical bonds is van der Waals interactions. It's the weakest of all the 4 bonds. Okay, it's more of a weak attraction more than a bond. It's actually an interaction between molecules caused by slightly positive charged or slightly negatively charged atoms. Okay, I describe this to my students more like since it's not a bond, it's more like I said, like an attraction. You know how when you get a balloon and you rub it really, really fast on your head and you have that static electricity where it kind of makes your hair stand on end? That's kind of like the attraction or the the weak attraction that van der Waals interactions is. It's very, very weak. Like I said, it's not really a bond. It's more like an interaction that occurs. So it's very, very weak. It's dipole to dipole forces that occurs between polar molecules. It's very, very weak interaction that occurs. So it occurs, and it's a dispersion force as well between nonpolar molecules like chlorine and chlorine. So very, very weak. It's even weaker than hydrogen bonds. So they kind of react and then let go, react and let go. It's a very, very weak bond, but they never actually hold on to one another. So I don't really consider this a bond at all. So it is the weakest of all of the bonds. So I kind of refer to this one more as an interaction than a bond itself, as far as bonds go. So water. Life began in water. It's the single most important molecule on Earth. All organisms are 60-70% water. Water has many unique properties that make it a life-supporting substance. Properties stem from the structure of the molecule, and the hydrogen and oxygen atoms within water molecules form polar covalent bonds. Remember what polar covalent bonds are. Okay? The electrons actually spend more time around one atom's nucleus than the other. What does water do in oil? Have you ever put water and oil together and try to mix it apart? Mix them up? Okay. Oil is nonpolar. Water is polar. That's why they will never mix. Okay? Okay? So, water will never ever dissolve in oil. Oil will never dissolve in water. Okay? Hydrophobic means water-fearing. Hydrophilic means water-loving. A substance that readily forms hydrogen bonds with water means it can dissolve in water as well. These are very important terms to understand, because when we get to talking about the cell in chapter 4, your plasma membrane has both of these features about it that's important to understand, because it is hydrophobic and it is hydrophilic at the same time. Yes, I did say it is hydrophobic and hydrophilic at the same time. That is considered amphiphilic. Okay, these are very, very important features for your cell to have, because it has to be able to control what goes in and out of your cell. Remember, your cell has DNA in it. Anything that goes in your cell can destroy your DNA. Guess what? If it messes up your DNA, it can cause cancer. You don't want it to do that. So very important what goes into your cells. So hydrophobic, hydrophilic, very important to understand. So why is water so unique and so important? Well, water stabilizes temperature. Have you ever noticed when you heat up water, it takes a long time. It doesn't just heat up instantly. A lot of other liquids will heat up a lot quicker than water. The reason being, those hydrogen bonds we just talked about, well, guess what? They sit there, they break apart, then get back together. Break apart, get back together. Break apart, get back together. Well, that is resisting the change in temperature that a lot of other liquids cannot do. That's why they have such a high specific heat. So that's why it takes a lot longer for it to actually raise the temperature of water compared to other liquids. Okay, they are most disrupted bonds than created ones as energy increases. This increase in energy has to be released through evaporation. Evaporation allows for the cooling of an organism through the release of individual water molecules at the surface. So, unique property of water. Another thing, as molecular motion decreases and temperature drops, less energy is present to break the hydrogen bonds between water molecules. That's another reason water can actually float when it's ice. Super, super cool property of water once again. A lot of other liquids will not float. water, good thing to know for lab. Water is an excellent solvent. What does solvent mean? Solvent means it will dissolve other solutes, okay? Solvent dissolves the solute. Solute is the actual substance that's actually being dissolved. So, for an example here, right here, this little molecule right here, which is your oxygen, your hydrogen, that's your water. So remember, oxygens and hydrogens, they have negative and positive properties, okay? So here comes your chlorines, here comes your sodiums. That's your table salt. So what happens if you put table salt or just salt in a glass of water? You mix it up. It dissolves, right? Reason being, your water sits there and breaks it apart. And as you can see, the hydrogens right here, which are positives, attach to the chlorines, which are negative. And the sodium, which is positive, well, the negatives, oxygens, attract and pretty much surround those guys. So everything that's the opposite of the other surrounds it. And so that disassociates it all. And like I said, dissolves it. And there you go. That's why water is an excellent dissolver or solvent. It is the guy doing the dissolving. So like I said, you're going to have to know this for lab. It's an excellent dissolvent or excellent solvent. Cohesion. So cohesion, a good way to remember it is cohesion is water attracted to water itself. So you know how, I'm trying to think of a way to do it. Have you ever seen those animals that are sitting on top of water surface itself and they don't break the water? Or that lizard that runs across the water that they call a Jesus lizard because it never breaks the water surface? That's cohesion. It's actually water molecules holding on and making that water surface tension, where water is actually clinging on and holding on with the hydrogen bonds, and it never breaks water surface. That's cohesion. Like molecules holding on to like molecules. And then there's adhesion, where water molecules, excuse me, water molecules are actually holding on to other molecules. A good example of this. Let's say you have a glass of water. You dump it all out. Can you ever dump out all of the water, every single molecule? Nope. You have little water droplets, little water molecules hanging on the side of that glass. That's adhesion, where there's an attraction between water molecules and other molecules. That those water molecules are hanging on the side of that glass. That's adhesion. Those adhesion and cohesive forces are important for water to actually be able to flow up the roots to the very, very tops of plants, so that all the highest and highest of those huge plants and national forests can get all the nutrients that it actually needs. Okay, this is an example of what I was talking about with cohesion. Okay, this is actually surface tension actually holding on, where this needle is sitting on top of this water and it's not breaking that surface. Reason being, those water molecules are holding on to each other. It's not breaking that surface, and it's actually causing surface tension of that water. Pretty cool. Okay, you guys see I made litmus paper in bold for you. Reason being, you're going to be dealing with litmus paper in lab. Okay, this is a pH indicator. It's going to tell you at what pH you're dealing with. Okay, so what is pH? pH is telling you the amount of hydrogen ions that's in that certain substance that you're dealing with. If it's a high number of hydrogen ions, that means it's a very low pH, or it's very acidic. If it's low number of hydrogen ions, then that's a very high pH, okay? Or it's very basic. Water is neutral, which is around a pH of about 7. Like I said, basic is less than 7.0. I'm sorry, acidic is less than 7.0, and alkaline is greater than 7.0. An example of blood in veins is around 7.4, and baking soda is basic, around 9.0 of pH. Okay. Acids are substances that provide hydrogen ions, which H is usually indicated by H plus right here, and lower pH. That's what acids do. They actually lower the pH. Because remember, lower pHs are acidic. So 2.2, 3.0, 4.0, anything below 7 means it's acidic. So the pH scale measures the amount of hydrogen ions in substances. So down here we have gastric acids, lemon juices, orange juices. So as you can see, we are actually able to drink orange juices and things like that, things that are very acidic down here. Reason being because our bodies have buffers that actually absorb that excess hydrogen ions, that is acidic ions, that are able to absorb those or those hydroxide ions and keep the pH of our body maintained. That's what buffers do in our body. Those buffers are actually readily absorb the excess hydrogen or hydroxide ions, keeping the pH of our body carefully maintained. Okay, our natural buffering system involves carbonic acid and bicarbonate ions. Too much hydrogen ions enter the body. That bicarbonate will combine with the hydrogen ions to create carbonic acid and limit the decrease in our pH. Too much hydroxides is introduced into the system, then carbonic acid will rapidly dissociate into bicarbonate, and hydrogen ions. Then the hydrogen ions can combine with the hydroxide ions, limiting the increase in pH. I put this up here specifically because in lab, you are going to be talking about this. So if I were you, I would take a picture of this, write this down, write it down in your lab notebooks. Basically, I want you to understand this because in lab, this will be a lot easier if you could write this out in a formula of some sort and understand what you're going to be talking about when you get to pH in this system right here. And last but not least, this is the last portion of this lecture. Organic molecules contain carbon, hydrogen, and may contain other minor elements such as nitrogen, oxygen, and phosphorus. Okay, a monomer is a molecule that is able to bond to other molecules to actually form a polymer. Okay, that's like how we were saying atoms combine together. So an atom plus an atom makes a molecule. Same thing. Monomer plus a monomer makes a polymer. Very simple. That's all it is. Monomer, make a polymer. Okay. A polymer, also known as macromolecules, means many monomers. That's all it is. Okay, just a different way of saying it. Okay, and the ones that we're going to be talking about in chapter 3, we're going to get into a lot more details about them, and that's what I was talking about when we talk about nutrition, what you're eating, what you're worried about what you're eating, should you be eating it, and so forth. That's when we're going to get into the details as far as carbs, lipids, proteins, and nucleic acids, which is DNA and RNA. These are some examples of those macromolecules we're going to get into in chapter 3. And if you have any further questions, email me, let me know any comments, concerns, and I'm here for you guys. Let me know anything. Like I said, you do want to know about the litmus paper for your pH indicator for a lab, and you are going to want to know about that pH in regards to the hydrogen ions and hydroxide ions for your lab as well. If you understand that, you'll be golden for lab. Good luck, you guys. See you later. See you next time on chapter 3 when we get into the good stuff. See you next time.